Jarrett today from, so she's coming, maybe I'm mixing it from Santa Barbara in Texas.
I work for UT Austin with Liz.
So from UT Austin.
Yes.
And, but she now lives in California. And she did her PhD at Florida State University. I just looked up because I took your CV so that you have quite a long list of awards, including a best paper in mathematical medicine and biology journal, which is a theme that a lot of us are interested in. And so we are pretty much look forward to your presentation today.
Okay, thank you for having me. So today I'm going to share with you some work that I've been doing the past couple of years. So this is all focused on breast cancer research. However, my graduate work was looking at primarily immune response and host response in persistent and resistant infections. So I've got quite a bit of a biomathy background, but all of the themes are still mathematical modeling and some analysis that we can apply to those things. So without further adu, let's motivate our talk. So this is something that this slide is very similar to what I showed the radiology department, but we have different questions when we're at Mathland. So why model cancer with mathematics? And I actually asked the radiology, why model anything with mathematics? Because unfortunately, not very many people like math as much as the people in this room. But it's a legitimate question. Why are we modeling this? What is the point? What can we do to actually help with this application? We shouldn't be modeling for the sake of modeling, right? Well, in cancer research, there are specific challenges. For example, we have reproducibility and repeatability issues, which you've seen recently in the news, unfortunately. There's an issue of bridging scales. So how do we go from in vitro information to in vivo information, from cell scale to in an animal or even in a person? What about across experimental designs, taking the same experiment but collecting different data? How do we connect that? And of course, in cancer research, we are doing trial and error. We have clinical trials. So without a specific framework, that really limits what we can do to optimize therapy and treatment for an individual patient, let alone a population, right? That's a huge problem. So for me, my overarching goal generally is to use mathematical models and their analyses, constrain them with data, and try to optimize therapy. And I say that generically because I don't mean only for cancer. If we can optimize antibiotics, that's awesome too. So that's just, I'm trying to introduce myself here a little bit, trying to show you my motivations and what we're looking for. Today, you're going to see data-driven integrated approaches with mathematics, some PDEs and ODEs. That's primarily what I work with. Some proposed multi-scale models, so kind of moving forward where I see these models going. And a very common theme in a lot of my work is uncertainty and sensitivity techniques to try and drive our understanding of the biological phenomenon. Okay? Okay, so the way that this is split up is actually two different sections. The first is, there are two different sections, there are two examples actually of the work I've been doing. So this first section is actually a project that I helped build up with a professor at UT Austin, where she had a bunch of data for this in vivo mouse system, and she wasn't sure what to do next. She said, I have all these pieces and I have a hypothesis of what's going on, but she didn't want to take the next step because, as you know, experimental things are extremely expensive. So, especially when you're working with animal models where you have to, first of all, grow them up and you have to implant them and do all of this extra work. So she wanted some guidance and was hoping math could do that for her. Okay, so the overarching goal here, this specific project, was to take this data that was tissue level information and try to use it to better understand the interactions going on for targeted therapy for a specific kind of breast cancer called HER2 positive therapy. Now, I gotta orient everybody in the room because it's very bio, right? So what is this data? What am I saying? What are these words? So HER2 positive, BT474, this is a cell line that's human derived, and it has a specific receptor on it. So this is how we recognize these cells. And these are breast cancer cells. And what happens is they take these cells, they implant it into a nude athymic mouse. What that means is the mouse has no thymus, athymic. So it doesn't have an intact immune response, but it has part of it. which is actually kind of nice from the mathematical perspective. I don't have to deal with the whole immune response, which is extremely complex. It's just the innate part, which is a lot of myeloid cells, macrophages, neutrophils, these really, some of the cells that we better understand in the immune response. So we implant those, and then these mice are treated with trastuzumab. What's trastuzumab? Trastuzumab, or TMAB, as we often say because it's a mouthful, is an antibody that targets the HER2 receptor. So what happens is it binds to it, and that binding will decrease stimulation of this HER2 receptor, but it also causes a signaling cascade in the cell. Okay? And that signaling cascade will reduce proliferation and also cause the cell to die, in some cases. So, reduces proliferation, reduces expression. Great. So this was all expected when Anna was doing all of these different experiments that she saw. She saw these tumors decreasing, but she observed something else that was completely unexpected. Vascular stabilization. What is that? So in tumors, if you don't already know, the vasculature is a mess. It's completely nasty. And the reason for this is the tumor cells are producing all of these pro-angiogenic factors. Because it's saying, I want vessels everywhere, I'm trying to grow. But this is totally unregulated. So what happens is you get these really immature vessels that are really leaky and they get crushed by the growing tumor. So it's really not normal vessels. So with the trastuzumab treatment, she was very surprised to see that this vascular stabilization. There was no reason for this to happen that we understood. So she had very, sorry.
What does it mean to say that you have vascular
Oh,
stabilization?
okay. So what was happening is when she observed the vessels after treatment, they matured, they stabilized, they became larger vessels, they did not collapse as easily under the pressure, and the way she quantified this, I'll show you in a second, is using MRI data. So you can really see quantitatively the change in this vasculature, okay? Okay? And so she had very specific hypotheses about this, and she thought it had to do with the immune response. Okay, so that's where I kind of go, oh yay, immune response. Do you think it's immune response? Let's work on that. So she wanted to work with a model to see and test her ideas. So this is basically a timeline of what the experiments look like. So we have tumor implantation. So inject tumor cells, they let them grow for 4 to 6 weeks until they reach a particular size. Then that mouse is considered on study. Once it's on study, we start to do the experiment. And controls receive saline, and the treated tumors receive the trastuzumab treatment. And then various different data are collected. So we have caliper measurements, F-MISO PET imaging, MRI imaging, histology collected. But the issue with this is all nice and clean and compact, but what really happened was a lot of different experiments, like I said earlier. She couldn't actually do a histology experiment and also a DC MRI experiment at the same time, because if you don't already know with histology, what happens is you take the tumor out, you slice it up and put it on slides and stain it and look at it. You can't use that tumor in the future. So it has to be a bunch of different experiments. Or sometimes when you do stains, you can't stain for one thing and another thing at the same time. What does this data look like? So first we have tumor volume. This is the easy one. They use calipers. I could do this. Go in the lab. You measure the tumor. You say how big it is.
I couldn't.
You couldn't do it. You probably don't want to. That's for sure. The other thing is something called dynamic contrast enhanced MRI, DC MRI. This is a gift, so it's going to repeat. But what happens is a contrast agent is injected, and then that contrast agent goes into the vasculature, and it will actually enhance the tumor and all of the vasculature within it. And there are specific models that have been well established in the MRI literature to quantify what's going on with the vessels. And so we could then, extrapolate from that the fraction of well-vascularized tissue for the model. Not yet. So this is H&E staining, so you can use this to identify your necrotic regions, so your fraction of necrosis. And then this is what I was saying, this is similar, this is F-MISO PET, which actually happens where they inject a radiotracer, and this radiotracer, it goes to hypoxic areas. What you're seeing down here, I think, is either like the bladder, so it's just filling up as it filters out, the very bottom part, but the tumor itself is being highlighted there.
So what is the scale? This is the whole mouse?
Yeah, this is a mouse that you're looking at. So the way that these mice are, the tumors are implanted, so they've got 4 legs, so it goes on their back haunch, basically. So that's what you're seeing here, and so you're looking down the mouse this way, where the tail's on our side and the head's on the other side.
So if it's a, I don't know how big a mouse is. But
They're very tiny. They're really, really small. They
so
fit in your.
what is the resolution here? 1 millimeter?
Oh, I don't know what she had for this one. I would assume, yeah, it has to be probably very small. For the, okay, so for the mice, for animal studies, you can do much better resolution because you can stick them in there for a longer period of time. When we get to the clinical stuff I'm talking about, it's usually a much, not higher resolution, a lower resolution because we have to get the patients in and out quickly. But yeah, I think it's probably one. I can look that up. I'm not sure what the resolution is for this one.
Why do you use PET specifically?
So this is what she was using. There are other ways to identify hypoxia now, and she's looking into this actually using OE MRI instead. So OE is oxygen-enhanced MRI to try and better identify these hypoxic regions, but this is just what she was working on at the time, the data that was available. And like I said, there's radio tracer is very specific to the hypoxic region, so that's what she was looking for. And then we have, so this is a comparison, just some examples between control and treated. So on the top, you have DC MRI, and you're seeing one of the parameters that you can derive from that data. So you can clearly see from the control to treated that, at least in these two cases, that there's much more perfusion of the contrast agent on the treated. The second one is the standard uptake value for that radio tracer. So you can see there's much more hypoxia for the control. And then the H&E staining here, you can see where we're identifying different regions of necrosis. And this is all pretty, and in math land we think, ooh, PDEs, spatial information, this will be great. But no, because we have histology, we're taking down tumors. We don't have one tumor where we have all of this information in time. That would be wonderful. What we really have are cohorts of tumors that we average, so this is going to be an ODE system for now. So I have the data here so that you can kind of look at what's going on. So the top left corner, can you see my mouse when I do this, or is this helpful? Yeah. Okay, so top left corner we have the control that's just growing and treated that's shrinking. So that's good, targeted therapy is working. On the right, this again is a DC parameter, so you can see that for the treated tumors this is increasing, and K-trans is the rate of perfusion essentially. And so here we have the controls that are actually decreasing a little bit. This is the F-MISO in the bottom left. So the PET imaging, so you can see hypoxia remains high, whereas for the treated tumors, they come down. And then that final panel here, there was no significant difference between the control and treated for the fraction of necrotic tissue for any time point. And I'll actually come back to that toward the end of this. That's an interesting fact that happens. So we had all this information, and then I'm kind of skipping this one at the bottom here, the fraction of immune response, because we didn't have this. This data did not exist when we started. But this is something that we will use to try and verify what the model was doing. So what's the proposed model? So this was a long process of Ann and I rapidly discussing all of her understanding of the different interactions of these different components and trying to write down a very simple ODE system. Without adding too much dynamics because there's just simply not enough information to know what all of these things were doing. So a lot of back and forth, trying to figure out what her basic intuition about what's going on in these experiments into the model. So I have a schematic here. I think we all love the schematics because you can kind of see all the pieces. The bottom four pieces, this is the information we have. We have the tumor volume, we have fraction of well-vascularized tissue, fraction of necrosis, fraction of hypoxia. Immune response, this is the part that's totally hypothetical, and we're just trying to see what happens. Okay? I'm just going to step through a couple of these just so you can see how direct and simple this is. So the tumor component, this is the equation. A few of you have already digested it, but the first part is actually exponential growth. So for the timeline of these tumors, and actually if you just let these tumors grow, they have no carrying capacity. They just keep growing. You have to take down the mouse. Eventually, it's that kind of tumor. There's also growth enhancement due to hypoxia. So this particular cell line, they become more and more aggressive with hypoxia and will grow faster. And the last piece is just death due to an interaction between the immune response and the tumor. So this is part of our hypothetical. We think that the immune response must be targeting this tumor somehow and enhancing this targeted therapy, which is from the literature. This is actually, some people have seen this.
So
yes.
T is the volume?
Yes, the tumor volume, and the other pieces are fractions of that tumor volume. The second part, I'll just show you the immune response. It's a little longer, but what you really have here are just two terms that are recruitment. So we have recruitment from the vasculature sending signals, and then recruitment from necrosis, which is actually one of the primary jobs of the immune response, to go in there and remove necrotic tissue and clean things up. This is maximally limited. And the last term is kind of cool. I kind of knew about this, but I didn't know we would see that in this system with just the innate immune response, non-adaptive, is that the tumor volume, these tumor cells, will produce all of these chemical signals that are actually trying to avoid the immune response. They send out anti-inflammatory signals to try and say, we're not here, don't detect us, nothing's happening, don't worry about it. Actually, some tumors are known to produce certain signals that recruit the immune response to help be pro-tumor, to help the neoplasm grow. So this is a piece that we have here, this reduction of the immune component. And we could walk through all of these pieces, and if you want to do that afterwards, that's great, but there's just not enough time, and I'd like to show you the cool stuff. So I'm going to keep moving. So what did we do with this model? We took the model, and we had the two different data sets. Luckily, we had enough information to calibrate and then validate. And so we calibrated using the controls and the treated, and we got two different results. So I'm going to zoom in on each of these, but to orient you, you got the control and the treated. On the right side, we have the tumor volume. Left side, the other components of that tumor. So here, for the control, you can see on the right, we have the simulation as that solid line. Then we have 95% confidence intervals based on the initial condition. And then the error bars are the actual data from the experiments. So on the left side, you can see the other components, but the one I want to point out to you is that immune response one, the blue line. The blue line is interesting because there's no data there, right? So we have no way of knowing if this is right or wrong at the moment. And you can see for the control, it kind of increases and then stagnates.
So can I ask
Yes.
a question? So you might have said it, but I just want to make sure I understand. So the stochastic component is because they have different initial conditions?
Right, there can be a range of when they start on study, basically.
That's the only thing
That's the only difference there. Yeah, that's where the 95% confidence is coming from.
Can I-
In this figure, yes.
So the control, when you give them Medicare, what does it do with it stops the signaling from HER2?
Oh, okay,
From
so
her positive?
the HER2 positive cells, the controls, they're given saline, which is nothing. So these cells just are growing like they would normally. Whereas the treated guys are getting that targeted therapy, which should disrupt the proliferation of these cells.
Because it stops the signaling.
Exactly. It stops that part of the signaling pathway. Of course, they're still kind of proliferating. It's not
All right.
100% perfect. Otherwise, the treatment would work.
What's the sample size here?
So different data points, different sample sizes. For I think
for the
for this,
initial conditions, I guess.
oh, for the initial condition. So usually there was, I think, in the entire cohort across all of the different experiments. Because remember, we have different data here. For the tumor volume, you have about 150 mice. Whereas for these, this is when things start to decrease. And as you get to the end time points, you get lower and lower cohorts of mice because you had to take some down. Good?
It looks like, I mean, the standard deviation is increasing over time, and for the real data.
Yeah, and that makes sense.
Aren't you missing a noise component
In the model?
Yeah.
That's a good point. So I'll show you some uncertainty graphs in a minute. That has to do with, we kind of looked at that from the calibration perspective. You're getting ahead of me, so hang on. I'll show you. Yes, I agree. So basically the model predicts that the controls are increasing and kind of stagnating for immune response. And for the treated case, it's totally different, right? Where the immune response is practically linearly increasing over time. Okay, that's pretty distinct. The model's saying one way versus the other. If I go back and forth, it's saying it should be significantly different by day 3 or 4, right? But how much confidence do we actually have in this? That's a legitimate question, right? You know, we have these error bars, what's going on? What we have is uncertainty. This is uncertainty. So for those of you who know what uncertainty is, great, but if you'd like a little mini review, there's two kinds. We have reducible and non-reducible. Reducible means we just don't have enough information, we don't know the parameters, kind of thing. We can reduce this uncertainty. For non-reducible, that means there's something fundamentally wrong with the model. We have to change the model to try and move forward. But in general, uncertainty just means that you have a lack of confidence in what your model's saying. I don't know if this is right. And you can look at it in many different ways. There's really fancy stuff. I love some of these names, polynomial chaos and fuzzy sense. But you can really look at uncertainty in very simple ways using just scatter plots and histograms. And that's kind of what we wanted to do, to just see if in our parameter calibration, do we still get a unique result here? So the idea is that we calibrated the model for the control and the treated. We did a calibration verification where we took the model, generated a bunch of in silico simulations, and then saw how well the calibration could actually recover those parameters. So we had error attributable to every single parameter. So the question was, okay, if I vary my parameters globally within their errors for the control versus treated, 1, does it still agree with the data? And 2, do we still get unique results? Or are they just going to be spread all over the place? Do I still get something different for my immune response? So that's what this picture is. So I can orient you here. It's kind of pretty. I thought it was fun. A lot of people look at it and go, whoa, what did you do? So what we did is we took the model, and just like I said, for each individual set, we varied the parameters within their errors, the known errors from our calibration verification, and globally sampled from there. And said, okay, what's the simulation? And we get, what's the simulation? And we got heat plots.
So sorry.
Yes.
So how many parameters are there?
Total, I think it was, I'm trying to remember. It's got to be 14-ish. It might be 1 or plus or minus 1.
So this is a 14-dimensional space. It's a horrible thing to sample.
Yes.
Okay. So, but you sample somehow in there, and then what you do with this sampling in the 14?
So then, from each of those parameter samples, basically, we simulated the model forward,
Okay.
and we basically counted up over time, what the values of the model were. So let me show you the picture. So what you hear on the left is control, on the right is treated. So this is way simpler than it needs to, it looks basically. On the top we have tumor volume, immune response, vasculature, necrosis, percent hypoxia. Black means there were no counts.
We're seeing like
It's like a histogram.
1,000 simulations.
Yes, so actually the number of simulations I think we ended up doing was around 2,000 total for each set. So what you're seeing, white is the hottest point, so the most counts. Black means none. Time here is increasing from top to bottom, so you see days, 0, 1, da, da, da. The red color is those 95% confidence intervals of the data, so that we can compare what the model simulated within its errors to the actual experimental results. Okay, so on the top we can see for control it looks really good actually. For treated it agrees. There's definitely places of disagreement and improvement that can happen in the model. That's expected, right? There's always room for improvement. But for the immune response this was nice. So when you actually look at the two of these, we still get these distinct results between the immune response. It's saying that, okay, for the control we should see less versus the treated we should see more. So at this point we said, okay, this is what the model's really saying. We should probably look and see, if this is a real thing. And that's all really Anna needed. She had a hypothesis. She needed just something to go off of to say, yes, let's invest some time and effort to see if the immune response is involved. Okay, so what do we do? Validation. This is immunofluorescence data. So this is from day 4. She had some specimens for control and treated tumors at day 4. And what happens is these immunofluorescent tags are targeted to specific things, so specific targets. And they go in and you can fluoresce them for different colors. Okay? So on the left side, you're seeing the actual image for controlled and treated tumor, controls top, treated bottom. And on the right side is some process, and I'm sorry that these are really quite pretty, but the projector doesn't want to show you how pretty they are. The process for 2 of the stains. So you can barely see it, but the top one is blue and it's dappy, and that's a nuclear stain. So this tells you viable versus necrotic tissue. CD11, CNF, or 80 are specific stains to a myeloid lineage cell. And if you have co-staining, this is an activated myeloid cell. Specifically, macrophages are in this column. So this is a really, if you don't know what a macrophage, you should Google it. They're really cool. And CD31 is a vessel stain. So the reason why we're looking at this CD11, CNF, or 80 is because we're trying to figure out that immune infiltration part. And on the right side, you can see we have the red and the green, and there's yellow. And yellow is the co-staining of these two. So if you just look at this example, you can see that there's definitely a difference, not only in how much staining is happening, but the way they're distributed in these tumors, which is really interesting. But what we really wanted to see was, were they different in the amounts? So it turns out that yes, there's a significant difference in the amount of not only just CD11, CNF, or 80 staining, but also in the co-staining of the treated tumors compared to controls. So this was really cool. We said, wow, all right, that's awesome. The model was actually right, yay! We were able to preliminary validate, at least for one time point, that we really should see more immune infiltration in these treated tumors. So that wasn't enough, though. We said, well, is there anything else we might derive from this? Is there any other information we might learn? Can we generate any other experimentally testable hypotheses? Because that's what it was. It made a prediction. We tested it. Well, in this model, we've talked about there are a bunch of different parameters, right? There's a lot of parameters. And when we calibrated, all the parameters look different. How do we know which of these parameters are important differences or not? So a few of you might have already thought, well, there's a method for that. It's called sensitivity analysis. So sensitivity analysis, if you don't know, is just a broad term, or it's just a broad group of methods that you can use to rank parameters by importance. What is importance? In this realm, in this definition, importance is if I have a parameter and I vary it just a little bit, and it causes havoc in my output measure, that is an important parameter. I need to know what's going on with this parameter. If I have a parameter that I vary a lot, and then I look at the output and it doesn't really do anything, okay, this is a parameter that's not so important. It's not really causing a huge effect. That output measure is very important. A lot of people will say, what is the value at the final time? But there are many ways to define the way you do sensitivity. So just because a parameter is unimportant and important for one output doesn't mean it is for all outputs. So very careful thing to watch for. I've found, in my experience, sensitivity can be used for lots of things. The main reason why we have it is to check model robustness and reduce uncertainty. That's the last point. But I've used it to use a lot for many different things. Identify important parameters for estimation. We can do a form of model reduction. This is a terrible way to say it, but really it's like a parameter space reduction. If you know a parameter is not very important in quotes, you can freeze that parameter and look at a smaller parameter space, depending on what you're trying to do. You can identify target parameters for model expansion. Say you have a parameter that's very sensitive. It might be that you need to expand that component so that you have more control. And also provide insight into the phenomena being modeled. So this is the part that I'm going to show you next, an example of that. There are two different kinds, global and local. I've only used global methods. But here's just kind of a list of a bunch of them. The primary one, the one I've used the most is Sobel indices, which is kind of the most widely accepted sensitivity use to measure. But I have used some of the other ones. So for example, PRCC, this is a nice statistical way to evaluate importance of parameters. And what's nice about it is if you have a really slow and clunky model, it doesn't really require as many simulations as, say, Sobel does. So you have to pick and choose what's most appropriate. There are some limitations to PRCC though. So it's one of those things where depending on what you're looking for and what model you're using, you may use a different method. So for this particular model, here are just some of the results. This is Sobel indices. These are total effects. Sobel actually gives you 3-fold information. Total effects are the variation of the parameter alone as well as in combination with other parameters. So this is kind of the most important it can be, any particular parameter. You'll see that there are different color bars. I think this is just the sensitivity of the output at the final time, what values it has. And so we have different outputs here, so you can see that different outputs can be sensitive to different parameters depending on what you're looking at. So I'm not going to go through this step by step, but using this information, we said, okay.
So,
oh, yes.
Sorry, I don't know.
No, it's okay.
Well, sensitivity analysis, but you have an ODE, it's easy to compute derivative respect to parameters.
So it depends on which method you're using. So Sobel is like a variance-based method, so using the ANOVA decomposition. PRCC is a statistical method that's looking at the monotonicity of the behavior. So depending on which method you use, yeah, it depends on.
But at the end, the sensitivity is captured by the derivative.
Essentially, for Sobel, you're looking at how much variation you get in the output at the end. It's not necessarily the change in the time, it's not a change, which it depends on what derivative you're.
Oh, derivative with respect to the parameters.
Yes, essentially, I guess that's one way you could look at it.
I'm
just being careful because sensitivity, people that work on it.
Still that you see how to have to solve.
I think that's actually how they look at it in dominant scale analysis. That they specifically are looking at those derivatives. It depends on the system and what you're doing.
But what you get is full Jacobian, and so it also illustrates the fact that some parameters are linked together. It's
Yes,
not-
exactly,
So
yeah.
does it make sense to study them separately?
That's a very good question. So if you have interacting parameters, a lot of people argue that the sensitivity is not really accurate. So depending on, so that's what's nice about solvable indices actually. So the threefold information you're getting is the total effects, which tells you what it's doing, as well as interacting with parameters, as well as main effects, which is its variation alone by itself. So there's something called additional effects, and I've looked at this in many different ways, and basically you're gonna have two parameters where all their sensitivity is coming from those additional effects, and you have no idea what that is. So in most cases, people say, oh, it's not appropriate, or oh, these are very sensitive, we just need to tack them down. People kind of just avoid that question. It's true in the literature. They kind of just try to skirt around it. I use that as an opportunity of, the model's just not quite right there. These should be separable. What do we need to do to change this model so that we can reduce that interacting effect? That's my approach. But that is a very interesting discussion that goes on in the literature for sure. What do we do with these additional effects? Okay, so where was I? Okay, so we have control and treated parameter sets. We now know which ones are sensitive. So is there anything we can learn about the phenomenon? So the first thing we saw was for the immune response in vasculature that we have sensitivity to their interactions, which is nice. This is part of what Anna was trying to figure out. Is there some interaction here for the treatment with trastuzumab? Sure enough, that popped out. Of course, we don't know for 100% sure if it's right, but you know, this is nice to see in the model reflected. This one was this is a little nuanced and kind of strange. We saw that there was a decreased deterioration rate of the vasculature due to the tumor burden. So remember, vasculature is the fraction of well-vascularized tissue. And we know the tumor is inducing angiogenesis, but it's also crushing things and causing havoc, and it's not good vasculature, right? So the tumor burden's already decreasing, so we should already see some relief on this vasculature. So why is the deterioration rate also decreasing? That's weird, right? Like, you shouldn't see both necessarily. So this suggests there might be something else going on here, and we weren't sure what to do with that. The last one actually threw us for a loop. This one was a weird one. So what we saw here is, remember, immune response, one of its primary jobs is to remove necrotic tissues. For treated tumors, the rate that the immune response was removing necrosis was decreased. Remember, there is more immune response in the treated tumors. And then it decreased its rate of removing necrosis. So when I first saw this, I was like, this is, I don't know, we might want to go back to the drawing board a little bit. This makes me uncomfortable, I don't know. But we went back to the immunofluorescence data, and it turns out that there was a significantly lower amount of immune infiltration in the necrotic areas for treated tumors compared to the controls. So this was really interesting. And remember that there was no significant difference between necrosis at any time point for the treated tumors compared to controls. So this suggests that there's this what's the word I'm looking for? Reprogramming of the immune response to focus on the other two areas, the vasculature and the tumor, and not necessarily necrosis. Remember, there was way more immune infiltration. There was plenty enough to go around in the treated tumors, but they are not focusing on the necrotic parts. And I can tell you, if I went back to the pictures,